Welcome back learners to Biotechnica's YouTube channel. Today we are breaking down complex molecules and metabolic pathways into bite-sized pieces. So whether you are a biology buff or a complete novice, get ready to demystify the magic of life at the molecular level. Let's dive in. Let me remind you that entrance exams having this topic in their syllabus common is CSIR NET Life Sciences, GATE BT Excel, ICMR, TIFR, ICAR and DBT as well. First, overview of the carbohydrate metabolism. In carbohydrates, the molecule which is used the most is glucose. Glucose is obtained from the nutrition that we take as food. It is broken down into the complex polysaccharides are broken down into the simple polysaccharide glucose, and it is absorbed into the bloodstream from the intestine. This glucose is picked up in all cells and converted into glucose 6-phosphate. The reason why phosphate group is attached to the glucose is to lock it inside the cell. Otherwise, the cell will be easily removing the glucose out of the membrane through the transporters. Glucose 6-phosphate, the phosphate in here adds a negative charge on this molecule, because of which it cannot cross the plasma membrane. Now, whenever glucose 6-phosphate increases inside the cell, it is an indicator of increased glucose inside the cell. That means intracellular glucose concentration, if it is high, accordingly it is decided which pathway it will be going to. Glucose 6-phosphate can be readily broken down to yield energy through the process of glycolysis. Glyco means glucose, lysis means splitting up. Breaking of glucose is what is glycolysis to a molecule which is called as pyruvate. Glucose is a 6-carbon molecule and pyruvate is a 3-carbon molecule. From one 6-carbon molecule, if you have to obtain 3-carbon molecule, then you will get 2 copies of it. From glycolysis, what is produced is 2 copies of pyruvate. This pyruvate can then be converted into acetyl-CoA, and acetyl-CoA enters into the citric acid cycle. From citric acid cycle, several reducing equivalents are produced, which can enter the electron transport chain and ultimately form ATP. This is beyond the scope of today's video. You can refer to another video regarding oxidative phosphorylation and photophosphorylation in another video in the Biotechnica YouTube channel. Okay. Comparing with the other metabolism, fatty acid metabolism can also yield acetyl-CoA through the process of beta oxidation, which is not that important from CSI point of view, hence we will not be covering it today. However, if you want to learn it, you can learn it in our regular Biotechnica classes. This acetyl-CoA, it is a central point of fatty acid metabolism, carbohydrate metabolism, and even protein metabolism. Now this pyruvate, it can have several fate. One is if you have aerobic conditions, then this is what it will give rise to, the citric acid cycle electron transport chain. But if it is in anaerobic condition, then the pyruvate enters into the lactate phase. It converts into lactate, which can come back into the pyruvate when again aerobic conditions will prevail. Now there will be some times when glucose source is not available, but our bloodstream should have glucose always, and this is controlled by liver to maintain blood glucose at least the minimum amount for use of the critical organs like brain, muscles, etc. If glucose in bloodstream gets depleted, the liver takes the responsibility of synthesizing new molecules of glucose. From what it will synthesize? It doesn't have sugar molecules in the blood. So it will be using its other biomolecules such as proteins and lipids, that is non-carbohydrate sources, and somehow it will channelize into the pathway called as gluconeogenesis to make new molecules of glucose. So gluco means glucose, neo means new, genesis means synthesis. New molecules of glucose synthesis. It is the reverse of the glycolytic process. Exceptions for a few steps, rest of the process is just reversed over here. Okay. But the last step, glucose 6-phosphate, only in liver cells they can convert into glucose to release into bloodstream. but not in other cells, because in other cells the glucose, they are trying to utilize the glucose and keep it inside the cell. If they have the reverse enzyme to release it outside, then it is of no use. So that is why only the liver cells, which are the master regulators of glucose concentration in blood, only they can convert into the last step. Another fates of glucose 6-phosphate, if they increase in concentration inside the cell, is storage. The polysaccharide glycogen, it is formed out of many molecules of glucose molecules. So the process of making glycogen is called as glycogenesis. Glycogenesis means glycogen and synthesis, okay? And the process of breakdown of glycogen is what is called as glycogenolysis, means glycogen breaking is what is glycogenolysis. Glucose 6-phosphate can also give rise to 5-carbon sugars, that is the ribose sugars, which are needed for the nucleic acid biosynthesis. Along with that, NADPH, also required for biosynthetic pathway, it is produced through this pentose phosphate pathway, from where again they will be shunted back into the glycolysis. That is why this pathway is also called as HMP shunt or hexose monophosphate shunt. Again, it is not important from CSIR point of view, so we will be not discussing it today. Over here, let's look at glycolysis, TCA cycle, gluconeogenesis and glycogen synthesis in brief. As we said, we are trying to simplify the metabolic pathways here. So we will not cover the regulation and any more further detail. We will just have a look at the basic flow. Okay. So glycolysis, it is splitting of glucose, which is a 6-carbon structure, into 2 pyruvates, which are 3-carbon structures. Total 10 steps are involved in glycolysis. You have the D-glucose present in the cell, which is 6-carbon. You can see this is 1, 2, 3, 4, 5 and 6-carbon. And it is converted into pyruvate. If you don't want to remember structure for exam, it is okay. But remember the details. Like pyruvate means it is pyruvic acid. So it will have an acidic component, that is COH. The dissociated form is written as CO-. And also it has 3 carbon. These are the details which you are supposed to remember. 2 carbons, 2 such molecules are produced out of glycolysis. The glycolysis process, the 10 steps are divided into 2 phases. So when we have glucose, each of this circle represents 1 carbon atoms, right? So the first phase is called as the energy investment phase, or it is also called as preparatory phase. As the name suggests, why it is energy investment, as 2 ATPs are required over here. The first 5 processes are the energy investment step. So from glucose you will be getting the splitting up of 3 molecules. So one 3 molecule is glyceraldehyde 3-phosphate. Another 3 molecule is the dihydroxyacetone phosphate. But that also again gets converted into glyceraldehyde 3-phosphate. So at the end of the preparatory phase, you will have 2 molecules of glyceraldehyde 3-phosphate. And from here the rest of the steps will be repeated twice. That means for each of the glyceraldehyde 3-phosphate. So from here onward, whatever you are requiring for the metabolic pathway or whatever is supposed to be produced, everything you have to multiply by 2. Okay. The next stage is the energy generation stage, or which is also called as the payoff stage. This is from the 6th to the 10th step. Here 4 ATP will be produced. That is why it is called as payoff or energy generation. 2 ATP in each of these steps. But since it is repeated twice, so there are 2 steps in which ATP is produced. But for 2 glyceraldehyde molecules, that is why 2 into 2 will be total 4 ATP will be produced. In one of the steps in this 6 to 10 steps, that means 6, 7, 8, 9, 10, in this one of the step, NADH is produced. Again, since there are 2 molecules of glyceraldehyde 3-phosphate, so total 2 NADH will be produced. Okay. Let's look at some more details of glycolysis. First is the preparatory phase. The 6 circles represent 6 carbons to make it simple. An important point in glycolysis you people have to remember is that all the intermediates will be phosphorylated. Remember, glycolysis is taking place in the cytoplasm. Until and unless pyruvate is made, we don't want any of the intermediates to walk out of the cell. That is why each of the components have to be phosphorylated, and only after pyruvate formation this can enter into mitochondria, where it converts into acetyl-CoA and can enter the citric acid cycle. So each of the intermediate of glycolysis, you can see a phosphate group is attached. We know that from steps 1 to 5, at the end of 5th step, we need to have 2 3-carbon compounds, isn't it? So individually they will proceed for the rest of the steps. So that means each of this 3 component, each of this part should have a phosphate group attached with it. That means 2 phosphate group needs to be attached in this first few steps. Okay. So first step of glycolysis is attachment of a phosphate group at the 6th carbon. If you start from 1, 2, 3, 4, 5, 6, the first phosphate will be attached at the 6th carbon. Thus you are making glucose 6-phosphate. Okay. Now another phosphate cannot be added just like that, because if you look at the structure of glucose, only 1 carbon is outside the ring. That is the 6th carbon where the phosphate group can be attached. Now phosphate group cannot be attached anywhere else. It can be attached only at a carbon hydroxyl group which is present outside the ring. Now you will see the rest of the hydroxyl groups are part of the ring. That means this glucose needs to convert into a structure which will have 2 carbons outside the ring. So 1 6th carbon which was already phosphorylated. Now another carbon will also be sent out. Now if you look at these 2 structures, this glucose has 6 carbons, 1, 2, 3, 4, 5 inside the ring and 1 oxygen. So this is what is a 5 carbon and 1 oxygen in the ring. But this structure which is having 1, 2, 3, 4 carbon and 1 oxygen. So this is what is called as a furan structure. This is what is called as a pyran structure. This is what we get in the fructose. That is why the glucose needs to convert into fructose so that another carbon is sent outside the ring for the attachment of phosphate. Okay. Hence, the next step in glycolysis will be conversion of the glucose to fructose. See, everything is easy when you understand the meaning behind it. So, glucose converts into fructose. Glucose is an aldose sugar if you know anything about carbohydrates, whereas fructose is a keto sugar. That means keto sugar means C double bond O, at both ends you have carbon, and aldose sugar means you have C double bond and you have hydrogen. So this is the aldehyde group. Okay. Now fructose 6-phosphate, another phosphate can be attached at the first position. So that is why you will have fructose. This is the first carbon 1. This is 6th carbon, 6th diphosphate. Di means 2 phosphate groups are attached at 2 different positions. Okay. And now this molecule is ready to be split off. So when this is split off, you already get the keto molecule, which is dihydroxyacetone phosphate. A 3-carbon keto sugar is dihydroxyacetone phosphate. And the other 3-carbon, it doesn't have a functional group, so it is converted into an aldehyde group. That is why the enzyme that you get, the enzyme that will be catalyzing here is what is called as aldolase. Okay. Now, talking about the other enzymes over here, the first enzyme here, they are adding the phosphate group. So, these are kinases. And since it is adding to a 6-carbon sugar, hence it is called as hexokinase. Then fructose 6-phosphate converting into fructose 1,6-bisphosphate. It is also a kinase, but it is adding the phosphate group to the fructose. That is why it is called as phosphofructokinase. Okay, so this is a major regulatory enzyme which is called as PFK1. Why 1? Because here the phosphate group is attached to the first position. Okay. And glucose to fructose conversion is just isomerization reaction. That is why this enzyme is called as phosphohexose isomerase. Okay, it is not adding or removing anything. It is just converting one form to other. And this is where I told you use the enzyme aldolase, as a keto group is already present, but you are making another aldehyde group. There can be interconversion from the DAP to glyceraldehyde because now the rest of the steps will proceed through glyceraldehyde phosphate. So this is again just a isomerization from keto sugar to aldose sugar. This is also done by an enzyme, but this is a different enzyme. This is also done by an isomerase, but the name is different. It is called as triose phosphate isomerase. Yes, enzyme names are very important, so remember them. Next is the payoff state. So conversion of the glyceraldehyde 3-phosphate to pyruvic acid is what is the payoff state. Reaction 6 to 10, as you can see, 5 steps are here as well. In the first step, we start with glyceraldehyde 3-phosphate. From here, 1-phosphate what was there. Now glyceraldehyde means it contains aldehyde. The aldehyde, that is CHO group, it is converted into acidic group. Acidic group means COOH. So acidic group, this is more oxidized, which means that loss of electron has taken place in conversion from aldehyde to carboxylic acid. Okay, those electrons, that means if they are lost, something should take those electrons. In this case, NAD plus takes the electrons and converts into NADH. So this is the step where NADH is produced. Since there are 2 glyceraldehyde 3-phosphate, that is why you get 2 NADH from here. Not only that, one more phosphate is added over here, because of which already at the 3rd position phosphate group was added. Now even at the 1st position also one phosphate group is added. That is where it forms 1,3-diphosphoglyceric acid. Now next step is where 1,3-bisphosphoglycerate will be converted into 3-phosphoglyceric acid. Why 3? Because one of the phosphate is removed. So this is a high energy bond which when broken will release a lot of free energy that can be used in the conversion of ADP to ATP. Since this free energy is coming from the substrate itself, that is why this step ATP production is called as substrate level phosphorylation as compared with the oxidative phosphorylation produced after the TCA cycle. So after losing a phosphate group, it becomes 3-phosphoglyceric acid. 3-phosphoglyceric acid, the phosphate group shifts the position from 3rd position to 2nd position, thus forming 2-phosphoglyceric acid. Now there is a removal of water molecule which results in a high energy bond called as the enol group, and thus the molecule will become phosphoenol pyruvic acid, or you can also say phosphoenol pyruvate. From phosphoenol pyruvate, again by the breaking of this high energy bond, free energy is released which is used in the conversion from ADP to ATP, and finally it will be producing pyruvate. The enzymes, since here we have oxidation reduction reaction, the enzyme will be dehydrogenase and since the substrate is glyceraldehyde 3-phosphate, so it will be glyceraldehyde 3-phosphate dehydrogenase. Here we have the formation of an ATP where phosphate group is transferred, so we call it as a kinase here as well, and the substrate name will be added over here, so it will be phosphoglycerate kinase. Okay, 3-phospho to 2-phosphate is an internal change, hence the enzyme will be a mutase. Again the enzyme is called as phosphoglycerate mutase based on the substrate name. In the next one, an enol group is formed, high energy enol group, hence the name is called as enolase, where water molecule is removed. So this is a dehydrogenation reaction and dehydration reaction. And pyruvate kinase, this is the last enzyme. Since ATP is produced here also, that is why it is called as a kinase. Okay. Next is gluconeogenesis. It is a synthesis of glucose from certain non-carbohydrate carbon substrates. As I mentioned to you, we have the glucose, 3 steps of it will be reversed over here. These are hexokinase, phosphofructokinase and also pyruvate kinase. So these 3 steps are irreversible in case of glycolysis. You can see that there are separate enzymes for this steps in case of gluconeogenesis. This direction it is gluconeogenesis and this direction it is glycolysis. Okay. So what are these enzymes? As I mentioned, gluconeogenesis is reversal of glycolysis. 3 reactions of glycolysis are irreversible. That means these conversions follow different steps and use other enzymes in gluconeogenesis. 4 enzymes facilitate glucose synthesis by this pathway by reversing 3 highly exergonic glycolytic steps. So from pyruvate, what we were getting phosphoenol pyruvate to pyruvate directly, in case of gluconeogenesis we get it in 2 steps. So from pyruvate we get oxaloacetate, which is a 4-carbon compound, and then again we get phosphoenol pyruvate, which is a 3-carbon compound. So 1 carbon dioxide is initially added, that is why the enzyme name is pyruvate carboxylase, and another carbon is then removed, which is called as PEP carboxykinase. You have to remember these enzyme names. The reason it goes into a 2-step process is because, remember I told you gluconeogenesis is the synthesis of glucose from non-carbohydrate source, and non-carbohydrate source like amino acids or fatty acids, they will make acetyl-CoA. Amino acids can make oxaloacetate. So these are to make them enter into this pathway as intermediates. That is why they come through oxaloacetate. Rest from PEP to fructose 1,6-bisphosphate, it is the same enzymes. Based on the equilibrium concentration, they reverse their process. And fructose 1,6 to 6, fructose 6-phosphate. In glycolysis, this was done by phosphofructokinase, fructose 6-phosphate to fructose 1,6-bisphosphate. But here it will be done by a phosphatase. So kinase is something which adds a phosphate group. So here they were adding the phosphate group to the first position. Phosphatase will be removing that phosphate group, converting back to fructose 6-phosphate. Thus this enzyme is fructose 1,6-bisphosphatase. Again, the fructose 6-phosphate to glucose 6-phosphate is done by the same enzyme. Glucose 6-phosphate now will be converted by another phosphatase, which is called as glucose 6-phosphatase. As I mentioned, these are only present in the liver cells to be able to regenerate glucose in order to release in the bloodstream. Next is glycogen metabolism. Here, 2 enzymes are very important. One is glycogen synthase, another one is glycogen phosphorylase. As the name suggests, synthase, it synthesizes glycogen. How do we start? Anyways, we start it with glucose 6-phosphate. Glucose converts into glucose 6-phosphate through the enzyme hexokinase. Now, glucose 6-phosphate instead of fructose 6-phosphate, for glycogen formation, it converts into glucose 1-phosphate. From glucose 1-phosphate, there is a requirement of attachment of a UTP. Generally, for such carbohydrate conversions, UTP is used. So UTP is attached with 3 phosphates. 2 of them is released out and 1 of them stays. There was one already present. So 2 of them makes UDP glucose. So this UDP glucose is necessary for it to get attached to the existing chain of glycogen. Glycogen is already a carbohydrate polymer, glucose polymer. So new glucose molecules will be added to the existing chain. If it is supposed to be added in the same chain, then the linkage will be made alpha 1,4. If it is supposed to be branched, then the linkage will be made alpha 1,6. However, this enzyme which will add the glucose to glycogen is glycogen synthase. Generally, this glucose synthesis will take place when there is excess of glucose. That means when insulin is there. We know glucose concentration increase means insulin hormonal signal will be increasing. So this insulin will be triggering the glycogen synthase. When you don't have glucose, so we will have the epinephrine and glucagon hormonal signaling. They will trigger the breaking down of glycogen. So glycogen in this case will be broken down into glucose 1-phosphate through the enzyme glycogen phosphorylase. So these will break the linkages that were formed by the glycogen synthase, and like glucose 1-phosphate can be again converted into glucose 6-phosphate. So these 2 enzymes, they are reciprocally regulated, and you are supposed to know all this for the exam. So further we have the citric acid cycle. It is very easy to understand citric acid cycle. So pyruvate converts into acetyl-CoA. The enzyme involved in that is pyruvate dehydrogenase. Remember, this pyruvate dehydrogenase is a very important enzyme. However, because of the time limit, I cannot go more detail into it. You can refer Biotechnica classes for more details. So acetyl-CoA, pyruvate was a 3-carbon compound and acetyl-CoA is a 2-carbon compound. So this pyruvate dehydrogenase removes one carbon in the form of carbon dioxide, and acetyl-CoA, it is a 2-carbon compound which enters a citric acid cycle. Since it is a cycle, there is already a molecule present before, which is a 4-carbon compound, and this is what is the oxaloacetate. A C4 and a C2 molecule links together to form a C6 molecule. 4 plus 2 is 6, and this molecule is called as citrate. Citrate makes a conversion into another isomer which is called as isocitrate, and this further proceeds. Now the next step will be to remove 1 carbon, because ultimately it should come to 4 carbon only, since this is a cycle. So from C6, 1 carbon is removed in the form of carbon dioxide, and it forms a 5-carbon compound. This 5-carbon compound is called as alpha ketoglutarate. Okay. Then 5-carbon compound again loses 1 more carbon in the form of carbon dioxide, and this molecule attaches one CoA molecule also, coenzyme A, and thus it will form something called as succinyl-CoA. Then in another step, that CoA molecule will be released, but that CoA molecule bonding was a high energy bond, which when broken yields free energy for the generation of GTP. So this is what is the substrate level phosphorylation for TCA cycle, after which it will generate succinate. Now succinate is converted into something called as fumarate. This is a part of the electron transport chain as well, and this is the only enzyme in TCA cycle which is present in the inner mitochondrial membrane. Rest all enzymes will be present in the matrix of the mitochondria. Fumarate converts into malate, and malate further converts into oxaloacetate. Okay. So in 3 steps, that means from C6 to C5 electrons were given to NADH. C5 to C4 also electrons were given. Even fumarate to malate, malate to oxaloacetate also electrons were given. But from succinate to fumarate, electrons are given to FADH2, because this is an enzyme complex present in the inner mitochondrial membrane, and it has an inherent FAD attached with it. That is why this is the only step where electrons are given to FAD and not NAD. That is why out of the TCA cycle, you will produce total 3 NADH, 1 GTP and 1 FADH2. Very important concept for CSIR exam. So these are the further details of the enzymes. As I said, acetyl-CoA 2-carbon combines with oxaloacetate to produce citrate. So since citrate is synthesized here without any ATP, hence the name of the enzyme here is citrate synthase. However, a water molecule is required here. But in the next step, the water molecule removes, as it has to convert into another intermediate called as cis-aconitate. But again the water molecule is added in a different way. So from citrate you get isocitrate, which is an isomer of citrate. Okay. Now from isocitrate, 1 carbon dioxide is removed, electrons are given. So you form alpha ketoglutarate. Since oxidation reduction is happening here, the enzyme name is dehydrogenase, and dehydrogenase uses the substrate name. In this case it is citrate. Alpha ketoglutarate again loses 1 carbon dioxide, and it will also give the electrons to NADH. So here also it is dehydrogenase based on the substrate name. Enzyme name is alpha ketoglutarate dehydrogenase. This also has a CoA which attaches with succinyl-CoA. In the next step, succinyl-CoA gives away the CoA part, but also releases free energy along with it, which makes GTP. This enzyme is succinyl-CoA synthetase as it involves ATP. Succinate is now converted into fumarate by that enzyme succinate dehydrogenase. Fumarate converted into malate by water molecule, addition of water molecule, but this is a stereospecific molecule, so always L-malate is produced. And malate converts to oxaloacetate, more electrons are released over here, so again NADH is produced. Since it is dehydrogenase enzyme, it's called as malate dehydrogenase. And here it is called as fumarase. The enzyme is fumarase over here. So that's all for as of now. I won't be discussing nucleotide biosynthesis here. Maybe it can be discussed in another video. Thank you everyone.